Welcome to BIOC211. This is Lecture 1, Part 2, and we're looking at Introductions to Cells and Disease. And today's lecture is going to be about cellular adaptation. So this slide is just a comparison of adaptive cellular changes with normal tissue. And we'll go through each of these individually throughout the lecture, but this is just so you can get an overview of what we're going to be looking at. And it's interesting to note that the only adaptive cellular change that can result in the possibility of altered cells migrating to other sites is neoplasm. So that's down the bottom right hand of your slide there. And this is associated with malignancy. So let's start with atrophy. So atrophy refers to the decreasing or shrinkage of cell size. So this commonly happens in skeletal and cardiac muscle and in secondary sex organs and the brain. So some of the causes are aging, lack of hormonal stimulation and HIV infection, and also disuse. So some examples of this are hormonal changes such as lowered estrogen levels in menopausal women can cause atrophy of the cells in the vaginal mucous membranes. Skeletal muscle, so lack of use of skeletal muscle and people who are bedridden, this will cause atrophy in those skeletal muscle cells. Now, cerebral atrophy is one that most of us are very commonly aware of, and this can occur due to brain injury. It can be caused by strokes or neurological diseases, so Alzheimer's disease, cerebral palsy, Huntington's disease, or even infections in the brain. And some of the symptoms of this are dementia, seizures, loss of motor control and memory, and difficulty with speaking, and also difficulty with comprehension and reading can be one of the common symptoms there. So just out of interest, that one of the best ways to improve cerebral atrophy or slow the progression of cerebral atrophy is to lead an active, healthy lifestyle, healthy eating, regular exercise, and increasing mental stimulation. So doing puzzles, things like crosswords, sudokus, memory games. And it's really important for the ageing population to be doing this, as this cerebral atrophy is seen in elderly people. And also just having relationships and conversations with people on a regular basis. So hypertrophy is the opposite of atrophy, and this is the increase in the actual size of the cells and consequently the size of the affected organ. This generally happens, or one of the common cells that this happens in is cardiac muscle cells and kidney cells. So this can be pathological or non-pathological, meaning that it could be disease-related or not. It can be caused by an increase in protein synthesis and a decrease in protein degradation, or both. And also excess workload can result in cellular hypertrophy. So a few examples for you are left ventricular hypertrophy, and this can be from hypertension. So that hypertension causes that cardiac muscle to become stronger out of necessity. It can cause aortic valve stenosis. So this is a pathological example. And this can be compared with a non-pathological example such as cardiac or skeletal muscle hypertrophy due to athletic training. So it's not necessarily an issue for athletes, because they need their heart to be strong and pump lots of blood around their body, and they need their muscles to be strong, and they need those larger cell sizes in those areas or tissues. But in some people, it can lead to a pathological symptom with cardiac hypertrophy, which can lead to damaged valves. So you can sometimes see this in endurance athletes over an extended athletic career, where that hypertrophy is so extreme that they can then have pathological symptoms due to that. So hyperplasia, this is an increase in the number of cells resulting from an increased rate of cellular division. So as opposed to the cell size getting larger, this is faster dividing cells, so you're getting more and more of these cells. As you can see from this little picture down below, they're normal cells versus the hyperplastic cells in the image. And this is common in epithelial, kidney cells and hepatocytes, and it is caused by increased mitosis, growth factors and hormones. So it is often seen in, this is benign prostatic hyperplasia, can result in, it is a result of her hyperplasia in those cells. And also the thyroid enlarges in pregnancy due to the stimulation of the pregnancy-associated high levels of TSH, or thyroid-stimulating hormone. And on this next slide, we have a picture of the cells in prostatic hyperplasia. As you can see, it's still glandular in appearance, but the cells are, but it is enlarged. There's just more of those little cells lined up next to each other. So next in the cellular responses is dysplasia. So this is often reversible. These are abnormal changes in the size, shape and organisation of the mature cells. So looking at your little diagram on this slide, you can see that the normal cells versus the dysplatic cells, they're sort of all just lumped in on top of each other, and they're all varying sizes. And as you can see in this little slide of a microscopic image of these cells, they're sort of a bit higgledy-piggledy. They're all lumped in together and all varied. So this can often happen in epithelial tissue of the cervix, and you may have heard of cervical cell dysplasia, and this is seen in abnormal pap smear results. Bronchopulmonary dysplasia is another condition. This is seen in premature babies born with underdeveloped lungs who require extended breathing assistance. And they can recover from this, often they recover from this, but not all the time. Metaplasia. So this is the reversible replacement of one mature cell by another, and is sometimes less differentiated cell types. So the best example I can think of with this one is Barrett's esophagus, which is often seen in clinic. Well, I find that I see this a lot in clinic, and patients come in with frequent acid reflux, and the normal cells in the esophagus, so the pseudostratified ciliated epithelium, end up eventually being replaced by columnar epithelium with goblet cells. And this is similar to the cells in the intestine. And this is what one of the causal factors is in Barrett's esophagus. So basically it's cells changing or moving from one place to the other and turning into different cells that shouldn't really be in that place. And often you'll hear after people have given endoscopy results will say, oh, the surgeon or the specialist said that I had stomach cells in my esophagus, and this is what they're talking about. Neoplasia. So this is, as we discussed in that first slide, uncontrolled progressive multiplication of cells, and this can happen in any fast dividing cells. Now, the causes of this are carcinogens, increased mitosis, loss of tumour suppressor genes that control cell division. So those tumour suppressor genes slow or inhibit that progression through specific stages of the cell cycle. And when these are impaired, there's no longer a checkpoint control for the proteins that stop that cell cycle if the DNA is damaged or the chromosomes are abnormal. And we can see this breakdown in the cascade of functions to inhibit cell proliferation, proteins that promote apoptosis, so that's scheduled cell death, and also the breakdown in DNA repair enzymes. So this is an example of the liver neoplasia. As you can see, there's abnormal clumps of cells in that liver image. Anaplasia. So this is the loss of differentiation of cells and their orientation to one another in their axial framework and blood vessels. So this is often seen in cancer cells, so usually fast dividing cells such as hepatocytes, so that would be liver cancer as you saw in that last image. And the causes are the loss of control of the suppressor genes and the cellular differentiation process. So the cells no longer function as part of the tissue that surrounds them. And in normal cells, the growth stops when a neighbouring cell is reached because normal cells communicate with each other to provide the proper shape for the tissues. So without this communication, each cell in a tumour showing anaplasia will grow to much bigger than a normal cell and will only be very loosely connected to the cells around it. And As a cell undergoes mitosis, it produces cells that also display anaplasia. So it's a bit of a vicious cycle, and this is how a malignant tumour begins. Now, tissue organ responses to these changes in tissue types. Scar formation and necrosis. So necrosis is the death of the tissue. And the organ responses are hypertrophy or atrophy of the organ, as we discussed earlier, altered function of that organ, hyper or hyposecretion of products, and overwork, exhaustion, and organ failure. And this can also result in loss of function. Aging is one of the main causes of cell death. And this is due to the atrophy of the cells causing decreased function. So we may see things like poor muscle tone, reduced fibroblasts in the skin, and this has been shown to lower collagen production. So this is part of the reason why we get wrinkles and our skin starts to get thinner as we age. Also, exocrine glands such as salivary and sweat glands reduce in function. Apoptosis, so this is programmed cell death. Injuries, so cell swelling is caused by injury. So this is due to the failure of the sodium-potassium pump, and the cell also loses its ability to produce ATP, which has that carry-on effect of impacting the sodium-potassium pump. Physical trauma, so pressure or tearing of the tissue. This can be from an impact, it can be from a number of different causes. Also heat, cold and radiation, so physical causes of trauma to those cells can cause cell death. Biological pathogens, so bacteria, viruses, parasites, fungi. Metabolic causes of cell death include hypoxia, so this is the reduction in oxygen flow to those cells. Toxic buildups or abnormal metabolites in the body. Nutritional deficits, fluid or electrolyte imbalances, these all come under that metabolic cause for cell death. And last but certainly not least is chemical causes of cell death. And so underneath this chemical umbrella, we're looking at things like toxins, so a poison produced by a living organism, so this could be the bacteria, so endo or exotoxins. Also poison, so any substance that irritates, damages or impairs the activity of the tissues. Foreign substances, so getting a splinter in your finger or your toe can definitely cause cell death. Carbon tetrachloride, so this is a chemical that's used in dry cleaning, and this has been shown to damage the heart, the liver, optic nerve, and a number of other nerves. Arsenic, so this is rat poison. This can cause diarrhea, vomiting, cramps, convulsions, coma, and definitely cell death. And cyanide, this is one of those chemicals that we have all heard of, but thankfully we don't see too much. But this inactivates cellular enzymes that are responsible for all respiration. So it basically shuts down your respiratory system and kills cells very, very quickly. And that wraps up part 2 of our Lecture 1. I will see you shortly for part 3.